Okay, so we were talking about this idea of aging versus cancer. We said that, it's kind of remarkable when you think about it. We don't think about aging as being something weird because, after all, that's how we're built. And everybody's used to seeing people aging around them, and you're used to seeing yourself, your own body aging. So you never question it, right? But it is kind of weird if you think about it. After all, if when we were first born and we were young, we have things like, healthy skin, healthy hair, etc. Why is it that by the time we're really old, our skin is compromised, our hair is compromised? You might have thought before that you say, well, it's just wear and tear, right? Your hair goes through a lot of wear and tear, your skin goes through a lot of wear and tear. But now you realize, it can't be that simple, because even in our youth, the skin on the outside of our bodies is always being shed and replaced by new cells, right? And so that's still going on when we're old. So why is it, though, that the quality of those new cells when we're old is just not as good as the quality when we were young? And so remember, we looked at this picture of comparing the skin of an 18-year-old with a 76-year-old. And we said that here, buried in here somewhere, are the stem cells that are constantly replenishing the keratinocytes. So you have a nice thick layer of healthy living keratinocytes here. They make keratin, so eventually when they dry out and die, they leave behind this nice thick layer of keratin. In the 76-year-old, of course, there's still stem cells down here. But the point is that throughout the many decades of life, we are going to lose numbers of stem cells, right? And if we lose certain numbers of stem cells, those are not going to be replenished themselves. So as we get older, we have fewer stem cells to replenish things like our skin. And importantly, after decades of being around, those stem cells are building up mutations. Maybe not mutations that are bad enough to cause the stem cells to apoptose, but they do represent little glitches, right? And so that means not only are we making fewer keratinocytes or skin cells when we're older, but the actual quality of those keratinocytes is lower than the keratinocytes that we made when we were a teenager. Right? And so why is it that Mother Nature is okay with this? Well, we're okay with this because that whole idea of limiting proliferation, right, and encouraging apoptosis is how we resist forming cancer. So it's always a balance. We accept the damaging effects of age because those damaging effects are a byproduct of the mechanisms of constantly fighting and preventing cancer from happening in our bodies. This figure relates to this idea that lifespan is a huge risk factor in development of cancer. So remember, a mouse lives much shorter than we do, maybe like 3 years or something like that. So if a mouse's tissue develops a cell that gets a hit that causes it to start going down the transformation pathway, by the time that transforming cell actually starts forming a tumor, this mouse has already reached the end of its lifespan. So therefore, evolutionarily, there was not as much emphasis in a mouse's genome on fighting cancer. But we live maybe 30 times longer. So if this cell is in our body, right, we have many, many more years for that cell to kind of stumble along through this path of different mutations, eventually becoming a malignant tumor cell. And keep in mind that we're meant to live maybe 40 or 45 years if you kind of follow the evolution of our species, right? So the fact that we live now to 80 or 90 years really highlights how we are sort of going ahead of what evolution actually anticipated our bodies to have to worry about in terms of, like I say, the warranty on our cells and the components on our cells, how they are designed, how many years of functioning they're meant to handle. So as the tumor cells are mutating, we said that although mutations are random, of course, certain mutations will matter more than others, not only in terms of what genes the mutations are in, but whether those genes are actually more relevant to the particular cell that the mutation is happening in. So different cells in different tissues of our body rely upon different pathways. A nerve cell has different pathways that are emphasized in its normal functioning of being a nerve cell compared to something like a gland cell or a muscle cell. So that suggests then that cancers developing in different tissues might preferentially reflect a bias for different mutations to happen in sequence. If we said in colon cancer, we might expect, for example, loss of the APC tumor suppressor gene. After all, that's how the APC gene was first identified. It's adenopolyposis coli, or a tumor suppressor gene that influences the formation of colon polyps when it's lost. Then you might follow through hyperplastic growth proliferation. You might end up with some sort of hypomethylation event in DNA that ends up activating a lot of genes that should have been turned off. Maybe you then get a hit in RAS, loss of another tumor suppressor gene, and maybe the fifth mutation that's important here is loss of p53, leading to carcinoma. So because mutations are random, right, you can actually look at probabilities. So again, this data is all based upon looking at something like 1,000 colon cancer tumors and actually seeing which mutations not only are the ones that show up the most, but being able to sort of like piecing together like a little investigative mystery story, figuring out which ones probably happened first. So in 80% of colon cancers, you get loss of APC first, followed by mutations in KRAS, 35%, followed by loss of one of these tumor suppressor genes, maybe involving the 8q locus, and then a bulk of them then losing p53, right? So this pathway would vary for different cancers, but the take-home message here is that this particular pathway is seen a lot in colon cancers, because this would be the order of mutations that kind of gets you from point A, a normal healthy cell, to point B, a fully malignant cell, most efficiently. So keep in mind that tumors take many years to form, because when you have an initiating mutation, like this one cell here, all of these gray cells are normal healthy cells in this tissue, but this one gets a hit, this red mutation, it's going to take many years for that cell to get to the point where you have maybe like 1 million of them, right? Because with every successive generation, daughter cells are going to do the right thing and undergo apoptosis. So it's going to be very slow, maybe like 10 years or so. But maybe after 10 years, you've ended up with 1 million of these cells that all have the red mutation. And now one of them gets a critical second mutation, the green one. That means this particular clone now has an advantage, a proliferation advantage. So it begins to expand faster than its neighbors. Faster also means an increased mutation rate, especially if this green mutation was loss of a tumor suppressor gene or loss of something that has to do with a DNA repair gene. So then in a shorter amount of time, now maybe 5 years instead of 10, you've now built up a population that all have the same baggage to start with, the green and red, because they all came from this second mutation up here. But now they begin to individually develop their own third mutation, whether it's the yellow one, the black one, the blue one, or the orange one, right? And that you begin to see is going to start giving rise to a heterozygous or heterogenic population of cells within just one large tumor. You can imagine, for example, if this blue mutation really increases the proliferation rate, then you'd have way more of these cells now beginning to take over as the bulk of the tumor, where these other types of tumor cells are all still mixed in there, but maybe they're not growing as fast. We looked at different ways that our DNA can suffer from mutations. So just as a reminder, go back in your notes and look at these 3 different ways. So mitosis itself results in mistakes that are introduced in the genome every time your cells divide. Then because your DNA is in living cells, they're surrounded by all kinds of biochemical processes that could be damaging to DNA. So in particular, metabolic reactions that give rise to sort of partially processed intermediate molecules that are very reactive. And those might improperly bind or damage the bases of our DNA. Then we also have exogenous mutagenic factors, things that come from UV radiation or exposure to things, for example, different types of carcinogens that we might ingest. So this is a reminder that DNA mismatch repair enzymes like the MUS proteins, oftentimes they work by simply detecting the proper form of the alpha helix. So if you have a mismatched base here, let's say that this double-stranded DNA represents a newly replicated strand, newly replicated DNA. So one of these strands was the template and the other one is the copy. And you can see the copy, there was a mistake in it that was not caught by the DNA polymerase itself, by its proofreading. So now it's up to these mismatch repair enzymes to come along and detect them. And the way these work is they look for deformities in the alpha helical structure. The deformity of the alpha helix causes the proteins to be deformed. And when they become deformed, you would have different regions of the proteins that then become exposed so that other proteins can then interact with them and maybe excise, cut out the damaged part of the DNA, as well as replace the DNA with the correct sequence. A lot of the metabolic processes in our cells result in these reactive oxidation species, right, which can damage our DNA. So we rely upon molecules like glutathione to help neutralize those ROS. And we said that glutathione is added to these ROS by this important enzyme, glutathione S-transferase, that takes the sulfhydryl group of the glutathione, adds it to the ROS, right, as we see here. So now we have the glutathione hanging off the end of the reactive oxygen species. And then if that reaction happens again to remove the glutathione, you're left with two hydroxyl groups here, which are far more stable because the oxygen is very happy here, right? Each one has two bonds to it compared to this oxygen that you started with, right? Which is highly reactive and able to go ahead and maybe cause damage by binding to something else. So remember, that's another example then that glutathione S-transferase, or GST, can be thought of as a tumor suppressor gene. And it's not surprising that levels of GST are often lost early on in the transformation process. UV radiation causes damage by cross-linking the pyrimidines in our DNA. So here we have two thymines next to each other here, right? So here they are. And we said that if the UV radiation, you just think of it as energy that causes all the atoms to vibrate and shake. If the energy is strong enough, these two carbons might break apart and they want to rejoin with themselves. And normally they would, but if you have two pyrimidines next to each other, then you have another two Cs right here that potentially could be where improper bonds form across the two thymines here, right? So that'd be a cross-link. And then this base, or these two bases, are messed up because if something like an RNA polymerase comes along and tries to read this in order to make RNA from this, if this were part of, like, let's say an exon from a coding gene, it's not going to know what to do with this, right? So that's going to mess up the ability of that gene to be expressed properly. Mutagenic compounds often cause damage by adding adducts. So in burnt food, we have a lot of benzopyrene products like this here. Again, you have a highly reactive oxygen group here, right, that then might bind and form a link or an adduct with a base of DNA here, this guanine. And so if you look, the resulting deformity of the alpha helix is caused by this big bulky adduct being in there. So again, this stretch of DNA is compromised. And if you cannot remove the adduct, then you have trouble. So we ended our lectures talking about this idea of stem cells then having to be protected. So as we said before, stem cells are deeper in our tissues, away from things that might damage them. And as they proliferate, they do so only occasionally, because with every cell division here, remember, you're going to end up making two daughter cells. One of the daughter cells becomes this transit amplifying cell, right? And it's going to go on and make many, many, many copies of itself that eventually, each of which then differentiate into a highly differentiated cell over here. Right? But the other daughter cell from the one mitosis sort of maintains its stem cell-like property, right? So the important thing here is it's not like this cell on the cartoon has simply moved down here and it divides again, or it moves down here and it divides again, right? It divides, so the original cell is lost, and it's just one of the daughter cells sort of keeps going. So any mistakes during the mitosis is going to be now carried by this version of the stem cell and added to every time it divides by more mistakes. So that's, again, why it is that if this was a stem cell that's making skin cells when we're 18 years old, by the time we're 80 years old, it's going to be down here somewhere, right? And it's going to actually represent many, many additional builds up of small mutations over time. But at least this shows you then that the infrequent division of the stem cell is part of how it gets protected from building up mutations too fast, right? Whereas the highly differentiated cells are the ones that we accept will be suffering a lot of damage, but it's okay because they are designed to be gotten rid of relatively quickly. Okay.